We all know that horses only give birth to little horses, cows to cows, cats to cats, and thus for each and every type of the living creature. But why is it so? It is because of the DNA or deoxyribonucleic acid molecules that carry the genetic information generation after generation, or otherwise the hereditary information is passed on from generation after generations by the DNA molecules. Hence, it is the hereditary material in human beings and all the other organisms. Recombinant DNA or shortly rDNA technology is a technique that cuts and pastes the DNA sequences from two different sources together using enzymes. Improvement in science is so drastic that a DNA fragment from other sources or organisms can be harvested and combined using enzymes by this technology. Let us learn in detail about this technology. DNA can be found inside the nucleus of the living cell. Each DNA molecule tightly packed with proteins and found inside the nucleus. The packed DNA with the protein is called chromatin and which form the chromosome. DNA unwinds during DNA replication so that it can be copied. DNA also unwinds at other times in the cell cycle so that the genetic information can be used to prepare proteins for biological functions. If a DNA is found in cell's nucleus, it is referred to as nuclear DNA. The complete set of nuclear DNA in an organism is called as genome. DNA molecules are also found in mitochondria in human cells and chloroplasts in plant cells. The energy needed for a cell to function properly is generated by mitochondria and photosynthesis occurred in plants' chloroplasts. DNA sequence has instructions or codes to form a protein is called as a gene. The size of a gene might vary in a wide range. For example, from 1000 bases to 1 million bases. The genome size of a human is 3 million bases, includes 20,000 genes in 23 pairs of chromosomes. DNA sequences must be converted into messages to carry out the functions that can be used to produce proteins. Proteins are the complex molecules that do most of the work in the human bodies. What does rDNA stands for? rDNA is the recombinant DNA. Recombinant DNA is the universal name for taking a piece of one DNA molecule and splicing it with another strands of DNA molecule. Thus, the name recombinant. The recombinant DNA is also at times referred to as chimera. A new strand can be created by combining two or more different strands of DNA molecules. Combining the DNA of two different organisms is the most common recombinant process. Recombinant DNA is a technology in which the DNA of interest can be cut and pasted using enzymes. The recombined DNA sequences can be kept into vehicles known as vectors that transport the DNA molecule into a suitable host cell where it can be expressed or copied. The word recombinant means joining of two things and comes from the word combination. The term recombinant means two things that has been joining back once again. In a simple way of understanding the technology, DNA recombinant rejoins two DNA of different organism into a single DNA molecule. Also, rDNA refers to gene manipulation or genetic engineering because the original gene is being artificially changed and altered. The formation of new combinations of the heritable materials by inserting the foreign nucleic acid molecules into any bacterial plasmids, virus or other vector system as it admits incorporation into a host living thing or organism in which it does not naturally occur, but still has the ability to continuous propagation, is defined as gene manipulation or recombinant DNA technology. In the recombinant DNA technology, the DNA will cross the natural species barriers in which the gene of interest of one organism is being placed into another unrelated host organism, and it will have the capability to propagate a clearly characterized and relatively small piece of DNA molecule in the host organism. Like the mythical chimera, the rDNA molecules are normally prepared of material from two different species, and hence they are sometimes referred to as chimeric DNA. The recombinant DNA technology uses the palindromic sequences, and this leads to the yielding of blunt and sticky ends. The DNA sequences that are used in the development of recombinant DNA molecules can grow or develop from any organisms. For example, bacterial DNA may be joined to plant DNA, or fungal DNA may be joined to human DNA. By the chemical synthesis of DNA, DNA sequences that have never occurred anywhere may be created and incorporated into the recombinant molecules. Literally, any DNA sequence may be developed using synthetic DNA and recombinant DNA technology, and thus introduced into any of a very wide range of the living organisms. Recombinant proteins are proteins that results from the expression of the recombinant DNA within the living cells. The recombinant protein is not necessarily produced when a recombinant DNA encoding a protein is introduced into a host organism. The use of specialized expression vectors is required by the expression of the foreign proteins, and frequently necessitates the significant restructuring of foreign coding sequence. Recombinant DNA method differs greatly from genetic recombination. In genetic recombination, the former results are from artificial methods done in the test tube, where the latter results in a normal biological process that remixes the existing DNA sequences in essentially all the organisms. Molecular cloning is the process that is used to create the recombinant DNA. This is one of the most widely used methods, along with the polymerase chain reaction, that are used to direct the replication of any particular DNA sequence that is being chosen by the experimentalist. The basic difference between the two methods is that the molecular cloning method involves the replication of the DNA molecule with a living cell, where polymerase chain reaction replicates the DNA molecule in a test tube, that is, they are free of living cells. A cloning vector is required for forming a recombinant DNA. Cloning vector is DNA molecule that replicates within a living cell. Vectors are normally derived from the plasmids, ORI-V viruses, and relatively represent a small segment of DNA molecule that comprises necessary genetic signals for the replication process, additional elements for inserting the foreign DNA, identifying the cells that has the recombinant DNA, and expressing the foreign DNA molecule. For molecular cloning, the choice of vector depends on the choice of host organisms, the DNA's size that is to be cloned, and how and whether the foreign DNA is to be expressed. By using a variety of methods, the DNA segments can be combined. For example, Gibson assembly or restriction ligase or enzyme cloning. The cloning of any DNA fragments basically involves 7 steps in standard cloning protocols: choice of the host organism and cloning vector, preparation of the vector DNA, preparation of the DNA to be cloned, creation of the recombinant DNA, introduction of the recombinant DNA into the host organism, selection of the organisms that contains the recombinant DNA, screening for clones with the desired DNA inserts and biological properties. The recombinant DNA can be made by 3 different methods. The 3 different methods are transformation, non-bacterial transformation and phage introduction. In the first step of the transformation process, a piece of DNA molecule that is to be inserted into a vector is selected. In the second step, the piece of DNA is cut using a restriction enzyme, and then the binding of the DNA inserted into the vector is done with DNA ligase. The recombinant DNA molecule is identified by a selectable marker during insertion. When exposed to a particular antibiotic, the host cell without a vector dies and an antibiotic marker aids this process. A host with a vector lives as it is resistant. Into the host cell, the vector is inserted and this process is called the transformation process. Escherichia coli is one good example having a possible host. To take up the foreign DNA, the host cells must be specially prepared. Transformed hosts can be distinguished from the untransformed host by using some selectable markers. The markers can be color changes, antibiotic resistance and any other characteristic that can distinguish. For different applications, different vectors have different properties. Some of the properties are size, symmetrical cloning sites and high copy number. The non-bacterial transformation process is very similar to the transformation process as described earlier. The only difference is in non-bacterial transformation, it does not use bacteria, while the transformation process uses a bacteria such as Escherichia coli for the host. The DNA is directly injected into the nucleus of the cell that is being transformed in microinjection. In biologistics, with high velocity microprojectiles, the host cells are bombarded such as DNA coated on particles like gold and tungsten. The process of transfection is known as the phage introduction. This is similar to transformation process. Instead of bacteria, a phage is used in this process. The packaging of vector is done by in vitro. To produce phage plaques, those contain recombinants, this uses M13 phages or lambda phages. Using various selection methods, the recombinants created can be identified by the differences in the recombinants and the non-recombinants. The foreign DNA present within the recombinant DNA construct may or may not be expressed following the transplantation into the host organism. That is, without expression, the DNA may replicate simply, or also it may transcribe or translate so that a new recombinant DNA is produced. Particular changes to the host organisms may be made to amend the expression of the ectopic gene. Changes may be needed to the coding sequences in addition as well. To optimize the translation, to make the protein stable, to direct the recombinant protein to the proper extracellular or cellular location and to stabilize the protein from degradation. How does an rDNA work? When the host cell expresses the protein from the recombinant genes, recombinant DNA will work. Unless expression factors are added, significant amount of recombinant protein will not be developed by the host. Protein expression reckons upon the gene being surrounded by a collection of signals which gives the instructions for the transcription and translation of the gene by the cell. Examples for these signals are the promoter, the terminator and the ribosome binding site. These signals are present in the expression vectors. in which the foreign DNA is inserted. Signals are always species specific. In Escherichia coli, these signals must be Escherichia coli signals, since Escherichia coli is not likely to understand the signals of human promoters and human terminators. If the gene contains introns or if it contains signals which acts as terminators to a bacterial host, problems are encountered. This will lead to premature termination, by which the recombinant protein may not process correctly, be folded correctly, or may even get degraded. In the eukaryotic systems, the production of recombinant proteins usually takes place in the yeast and filamentous fungi. Some proteins are too complex to be developed in bacterium, so eukaryotic cells must be used. In various fields like DNA fingerprinting, gene therapy, vaccine production, commercial and pharmaceutical products, etc., the recombinant DNA techniques is used. A double strand DNA is cut at a particular point using a restriction enzyme to form recombinant DNA. A second piece of DNA is cut by using the same enzyme. The complementary ends of each strand will bind with the other strand when the fragments are mixed together, thus forming a recombinant DNA molecule. To increase the DNA quantity, a stable genetic change is brought by the uptake of the naked DNA, and this is referred to as bacterial transformation. The state of being able to uptake the exogenous DNA from the environment is known as competency. Two forms of transformation and competency. They are natural and artificial. The natural transformation is a bacterial adaptation for the DNA transfer that reckons on the expression of many bacterial genes whose product seems to be designed to accomplish this process. Naturally, transformation is an energy requiring complex developmental process. Bacteria should become competent in order to take up, bind and recombine an exogenous DNA into its chromosome. Competent refers to a special physiological state. Expression of about 40 genes is required for the competence development in Bacillus subtilis. The DNA that is integrated into the host chromosome is generally derived from another bacterium of same species, and thus it is homologous to the resident chromosome. The length of the transferred DNA is greater than 1271 kilobases in Bacillus subtilis. The length transferred is probably double stranded DNA and is more often than a third of total chromosome length of 4215 kilobases. In prokaryotes, the natural transformation appears to occur. In 67 prokaryotic species, this process is happening. Artificial competence is the laboratory process in which the cells can be made permeable passively to DNA and exposing the DNA molecules to the conditions that do not normally occur in nature. The cells are allowed to incubate in a solution containing divalent cations, mostly in calcium chloride solution, under cold conditions, which is further exposed to a pulse of heat shock. Even so, the mechanism of the uptake of the DNA molecule through chemically induced competence in the calcium chloride transformation method is not clear. Since the phospholipids and lipopolysaccharides are present on the surface of the E. coli cells, it is negatively charged and thus the DNA is also negatively charged. The purpose of the divalent cation is to shield the charges by coordinating the negative charges and phosphate groups, allowing the DNA molecule to adhere to the surface of the cell. It is also proposed that exposing the cells to the divalent cations in cold conditions may weaken or change the structure of the cell's surface by making it more permeable to the DNA molecule. A thermal imbalance is thought to be created by the heat pulse on either side of the cell membrane, which further forces the DNA to enter into the cells through self pores or through the damaged cell wall. For promoting the competency, electroporation is another method. In this session, we studied about recombinant DNA, its technique, the process and its applications. rDNA is the abbreviation of recombinant DNA. Recombinant DNA or rDNA is a technique that cuts and pastes the DNA sequences together using enzymes. The recombinant DNA is also at times referred to as chimera. The word recombinant means joining of two things and comes from the word combination. Recombinant proteins are proteins that results from the expression of the recombinant DNA within the living cells. Molecular cloning is the process that is used to create the recombinant DNA. Using three different methods, recombinant DNA can be produced. They are transformation, non-bacterial transformation and phage introduction. In the transformation process, a piece of DNA molecule that is to be inserted into a vector is selected. Non-bacterial transformation is similar to the transformation process. The process of transfection is known as the phage introduction. To increase the DNA quantity, a stable genetic change is brought by the uptake of the naked DNA, and this is referred to as bacterial transformation. Bacterial transformation is of two types. They are natural transformation and artificial transformation.